Thank you. First of all, I would like to thank the organization, the Fundación Contra Hipertensión Pulmonar, for inviting me to show you what we work here. Many of you are colleagues of, and we work together. So I would like to summarize, because we don't have enough time and we are in a hurry up. We all know the current classification of PAH, which is based on clinical features. So we know that there is a molecular and genetic backgrounds in idiopathic, irritable, and right now in pulmonary veno-occlusive disease with different pattern of inheritance. But this is really reflecting what is going on in the background of the patient. I mean, in the physiophysiological mechanism and in the development of the disease. So there is a current of researchers and clinicians that they think that it might be a better idea to classify patients based on omics technologies. And one of the initiatives is the PVDomics, which is currently led by Anna Emes in the Vanderbilt University Hospital in the United States. And they think that the molecular mechanism underlying PAH must be addressed by analyzing the omics technology, such as the genome, the transcriptome, or the proteome, on all different technologies, and they will give us a better idea on how patients from even different clinical initially groups can be grouped in one cluster because of the development of the disease. And this is growing really fast, based mainly in the development of new generation sequencing technologies, not only for DNA sequencing, but also for RNA or even for proteomics or macroRNA, as many of you have talked previously. So this can be done, and I'm not going to explain a bit further, because of the reduction of the cost of the next generation technologies that allows in the last year to describe a battery of several new genes that are involved in the progression of the disease. So at the beginning, and we share many slides with Richard, sorry about that, BMPR2 was the main actor in the PAH. But right now, the map is much more complex and includes many genes and many different pathways. For example, the KCNK5, which many of you have talked, BMP9, developmental processes such as TBX4 with the relation with the small patella syndrome, even the angiogenesis processes, which top BP1 has been described in 2016, or even genes related to FGFR2 pathways. This is highlighting the importance of the genetic background of patients with PAH. So in terms of how we analyze our patient in the laboratory and how we manage that, in order to get a diagnostic, specifically for idiopathic and for irritable PAH, in which BMPR2 is not explaining all the cases, in the research we have the genome. As a researcher, we can sequence the entire genome of one patient. But in that case, we will do it in a research manner, because the explanation and the interpretation of the results are much more complex. But in the diagnostic setting, and right now as a gold standard in many laboratories involved in the diagnosis of PAH, is to do exome sequencing, which represent only 1% of the genome. So we have to keep it in mind, because it's really important. We are only seeing at what percent of the entire genome, which is the coding region of the genes. But we can also do a gene panel, which means that we can select only those genes that are related with the disease. In this case, with PAH, which right now there is around 35 genes if we count the genes with higher and with the lower evidence related to the disease. But it is also important that to have in mind that genetic is not all in PAH. We have to have a good and a deep phenotyping, and this paper summarizes really well how we work right now in the clinical and the molecular settings, because once we, you as a clinician, have a patient in the consultation, it is important to have all the clinical parameters and the feature of the patient well established and well characterized, because we have seen that the progression of the disease is not the same if we have a mutation in one of the genes, for example, in TBX4, or for example, in EF2AK4, which is the worst form of PAH. So altogether, codified with a systematic ontologies will help us to get the correct diagnostic and to find into the correct gene. For example, in cases with consanguinity, which we have seen that veno-occlusive disease is the worst subtype of PAH, the clinical feature will allow us to focus specifically in EF2-AQ4, but for example, in patients with PAHs that develop it in childhood or in early age, we can see in another gene, for example, TBX4, in which we have seen that patients develop this at early ages. So in terms of research and what we do when we have a patient in which we didn't find any mutation or, pathogenic or likely pathogenic mutation in the well-known genes, so we started whole exome sequencing analysis by trio or by single programs, and it's because in most of the cases the diagnosis of the disease is done in adult age. So we are not able to get samples from parents. So we have to analyze in a single manner. So the second step is to do the segregation analysis after the prioritization of variants. So once we have some candidate variants, we go into the segregation analysis in order to see whether the disease segregates with other members affected in the family or are absent in healthy parental samples. Then we carry out some functional in silico characterization. This means that we put the genes in the genomic context and in the protein context of PAH regarding the pathways in which the disease are acting. And the next step is really important, which means that we have to connect with another researcher all over the world with different tools. For example, in this case, Gene Matcher, and we can find and we can search if there is any other researcher working in the same disease that may have mutation in the same gene in another family in order to combine and to aggregate more families with the same or with a mutation in the same gene. And final step, but also important, is to functional characterization, as Mauro tell us previously, is really important to finally validate in a functionally way the variant that we have been detecting in our analysis by whole exome sequencing. And I'm going to show you two of our preliminary results of two families that we carry out whole exome sequencing and in which we found interesting candidate genes, and I'm now presenting you in a primacy. In TNIP2 and TRAF2, these two genes has been detected after our prioritization in two families, one with heritable PAH in which the mother and the proband has the PAH and both has the same mutation. This variant is not present in the control population in which we analyze them, and is not present in the healthy sister and in the father. So it means that this variant segregates with the phenotype. We validate this variant by Sanger sequencing, which is a routine analysis in our laboratory. And in our second, and just to finish, the variant is a missense variant. So we will go in deep later about the possible mechanism for this variant and in the PAH. And the second family is an idiopathic family with a girl that has a nonsense mutant in TRAF2, which means that probably the mRNA will be degraded and there is no function for this mutant because cause premature stop codon. This variant is not present in the healthy parents and even not present in the general control population in which we have seen this variant. So what we done is to analyze based on in silico tools in order to see whether this variant can be predicted as a pathogenic. And if you can see here, we apply several pathogenicity predictors, and sometimes some of them tell us that the variant could be pathogenic, and some of them could tell us that the variant is not deleterious. So that's why it's important that the in silico analysis will be confirmed by functional assess. Both of the variants, as I mentioned, are not present in the general population. So this means that this is a really rare variant. They doesn't have any frequency in the general population. So after that, what we think in our mind is to correlate and to put these two variants in these two genes in the genomic context of PAH. So we started to see how they can act in the organism to develop the disease and how this can lead to pulmonary hypertension. And in collaboration with some researchers in Stanford University, we have developed an in silico model that can explain why AVIN2, which is the protein encoded by TNAP2 and TRAF2, can lead to pulmonary hypertension. And as we have seen, the main processes that are affected in pulmonary hypertension and that can be downstream of the mutation of these two genes are proliferation, survival, and apoptosis, which is the main cellular processes that are affected in the disease. So we have seen that both genes are involved in the same pathway, which is the NF-kappa beta pathway, and both can increase the proliferation and can also be involved in survival of the cells that can be explaining in somehow the remodeling of the vessels. And the apoptosis is downregulated by two different mechanisms. So it can explain why we have more cells with a mutation that can be doing the remodeling, the vessel remodeling. So in my mind, and just to finish with this slide, what I think in pulmonary hypertension is that we all have to work together in a collaboration manner, not only for genomic analysis, but also for transcriptomic and proteomic analysis, having in mind the diagnostic with the importance of the clinicians and the phenotype of the patient, how is the follow-up, not only, for example, for a patient with homozygous mutation in PVOD, but also for carriers, or for the low penetrance that Richard have talked in his first talk. But also to have in mind that the final step is to get into a therapeutic effect. So, for example, it means that we can, for example, do some kind of GWAS analysis in order to see whether we can find some biomarker that can explain why some patient responds to drugs and other patient doesn't respond before to have some problem like edema or something that we have seen in our samples. At a genomic level, we can do panel of genes, but right now in the clinical, in diagnostic settings, the gold standard is to do whole exome sequencing, and probably in the future will be the whole genome sequencing, which is right now only for research. And in terms of transcriptomic and proteomics, some of my colleagues showed that mRNA-seq are really powerful tool because we can also see for other underlying molecular mechanisms that at genomic level we cannot see, for example, splicing or overexpression of different genes, and finally in the proteomics level. And some of the conclusion I already mentioned before, this is a phenotypically heterogeneous disease, and it's essential the collaboration between basic research and clinician in order to improve the diagnosis and the treatment and management of the patients. We have increased our diagnostic rate by applying next-generation sequencing technologies, but it is necessary to do further experiments to confirm the role of the gene that we are finding, and an early diagnosis and genetic counseling will help us to manage the patient properly. And finally, the current PAH classification based only in clinical parameters does not reflect the idea and the underlying mechanisms that are suffering this patient with PAH. And just to finish, I would like to thank all the people, all the patients, all the hospitals that send samples, because this work cannot be possible without the support of most of the researchers, not only from here, but also from outside, and also from our sponsors that help us to do this work. And thank you very much.
Thank you, Jair.
Just specifically, in the two genes that you showed us, whilst those particular variants are not present in the databases, are there other mutations that look rather pathogenic in the databases for either of those two genes? And if so, what phenotypes do they have?
So you mean these two genes? If there is any other mutants in these genes?
Yeah, in the public databases.
Related to PAH or in general?
No, in the public databases.
Yeah, I mean, these two variants are not present in the...
It's other mutations within
With phenotype, weird phenotype.
And do they have anything?
No, I didn't see anything about that. So right now, these genes are not related to any genetic disease, and there is variants, but they are not classified as pathogenic.